we see how are these cells, what are eukaryotic cells and what are prokaryotic cells. Okay, now let me get something. Now with these eukaryotic cells, eukaryotic, it comes from a Greek word, or there are two Greek words, which means eu, and eu means true, and then karyotic. Karyotic means nucleus. Okay, there is no e here. Then prokaryotic, pro means before, or in this case you can say false. So karyotic, nucleus. So prokaryotic cells, you can say these are cells with a false nucleus or without a nucleus. And then eukaryotic cells, you can say these are cells with a true nucleus. And we are going to see examples of cells, and we are going to see the differences between these eukaryotic cells and prokaryotic cells and how they are able to exist. So all cells, both prokaryotic and eukaryotic, they have these features. Number one, they have a plasma membrane. All of them, they do have a plasma membrane, whether eukaryotic, whether prokaryotic. And it is this plasma membrane that works as a regulator of what is entering and what is leaving a cell. So you must have a plasma membrane. They all have that same fluid substance called a cytosol. So it is this fluid that it is embedded within a membrane, okay, and it is the one that has the organelles that are found within this cytosol. And it's where chemical reactions are going to occur, because imagine in senior one you used to draw cells like this, and you draw this as an animal cell and you put a nucleus, and then you label this as a nucleus and a membrane and a cytoplasm. So is this cytoplasm just space? So that's how senior ones, they lied to us. Me, I thought this was just space, and then I used to wonder, how does this nucleus then hang in that space? But this is not just space, but rather a complex array of organelles and cytosol. In fact, it is fluid filled. Okay? And we are going to see. This is a tour of a cell. By the time we finish this topic, we shall be able to know what a cell is. Then you have chromosomes which carry genes. Okay? And we are going to see that these chromosomes are found in a nucleus. That is if you are an eukaryotic cell. But if you are not, then there is some region where these chromosomes are found. And that region is called a nucleoid region. I hope that is the spelling. You can check for it. Then all cells must have ribosome. Now this is like a factory, an industry, okay, where proteins are made. And you know that at this level you must be knowing that proteins are so important in our bodies. All physiological processes, all chemical reactions that occur in our bodies are controlled by these proteins. So all cells, regardless of it being an eukaryotic cell or a prokaryotic cell, they all do have ribosome. But still they vary. Okay? The ribosomes are not the same. For example, you find that with eukaryotic cells, for them they have 80S. Okay? Whereas prokaryotic cells have 70S. Now this one is just, the S it means Vedbugs, and it is a unit It is gotten from the other centrifugation, the other process we studied. Okay, that is a speed by which a given organelle sediments. Okay, so with prokaryotic ribosomes, they sediment at 70S. That's why they are called 70S ribosomes. With eukaryotic cells, they do sediment at 80S. So that is the difference. However, they all do possess ribosomes. Now, the cells can be of two types, the prokaryotic and eukaryotic cells. Now, only organisms of the domain bacteria, okay, which consists of archaea and the eubacteria, they are the ones that belong to prokaryotic cells. Okay? And so by now, you should be in position to explain, to determine what makes an eukaryotic cell and what makes a prokaryotic cell. Is that okay? Yes, we have seen that all cells have this. All of them, they do have that. But is it the same in all cells? For example, you'll be able to find out that the plasma membrane of eukaryotic cells has cholesterol, but the plasma membrane of prokaryotic cells lack cholesterol. So you should be in position. Yes, they have, but is it the same? And what is lacking among the two cells? Then protists, these are unicellular prokaryotic, eukaryotic cells made up of one cell, but they have eukaryotic cells. Then fungi, animals, and plants, all of them do consist of eukaryotic cells, true cells, cells with a true nucleus. So let's talk about the prokaryotic cells briefly. Now these ones, cells are characterized by having, what do you base on to say that this cell is prokaryotic and this cell is eukaryotic? And if you know this, then you probably have a good picture of what a cell is about. So number one, they have no nucleus. Okay, cells totally lack a nucleus, and that's why they are called prokaryotic. And so since they have no nucleus, do they have DNA? Do they have chromosomes? Yes, they do. But unlike in your cells, the chromosomes are enclosed within a nucleus, within an organelle called nucleus. But these ones, the chromosomes are naked. They are just within a nucleoid region. If this is your cell, we are going to see a cell. Then you have a circular chromosome that is just within this region. Okay? And we call this region a nucleoid region, not a nucleus. Number two, the DNA is unbound. Okay? Is an unbound region called a nucleoid region. Yes, I've talked about this. So unlike the eukaryotic cells where you find their DNA in a nucleus, okay, these ones, the DNA is within that nucleoid region. No membrane-bound organelles. For instance, you cannot find a mitochondrion. You cannot find a chloroplast. You cannot find a nucleus. So these double membrane-bound organelles, don't expect them in prokaryotic cells. Okay? Why? Because the way they are formed. We shall study the endosymbiotic theory, and we talk about, so you'll be able to see that then they can't have, okay, they cannot have these double membrane bound organelles basing on the endosymbiotic theory. Number three, cytoplasm is bound by a membrane. So this one is almost the same as eukaryotic cells. So you have a cytoplasm, and the cytoplasm is enclosed within a membrane. So that is one of the characteristics. The 70S, I talked about this. Okay? Yes, they do have ribosomes, but the ribosomes they sediment at this speed, okay, in a centrifuge. And so these ribosomes are referred to as 70S. And we are going to see that for eukaryotic cells, for them now they do have 80S. So that difference, okay, is remarkable. It's a remarkable difference that we cannot neglect. And the S stands for Svedbergs. Okay, this is this is a unit that we use to measure that speed at which a given organelle is going to sediment. Now, unlike plant cells whose cell wall is made up of cellulose, now these ones, their cell walls are made up of a conjugated carbohydrate, okay, called peptidoglycan. Now, this one is a carbohydrate, okay, it is a carbohydrate with a short chain, okay, of amino acids, or with a short peptide chain. Unlike in plants where it is purely cellulose, and cellulose is 100% carbohydrate, okay, but they all do have a cell wall. So when you are giving similarities, so this one can work as a similarity. You are comparing a plant and an animal cell. And this one works better as a similarity. They both do have a cell wall. Okay? However, in prokaryotic cells, the cell wall is made up of peptidoglycan. Whereas in eukaryotic in plants, the cell wall is made up of cellulose. About fungi, yes, it is chitin. We can say chitin, depending on the kindergarten. So basically, that's how it is. This is a genome structure. Okay? So you can see this is the circular DNA that we are talking about. And the DNA is enclosed within a nucleoid region. Okay, it is just embedded in a nucleoid region. So this is our nucleoid region. So the whole of this is a cytoplasm. Where is it? Yeah, it is labeled here. So the whole of this is a cytoplasm. Then these ones, these tiny particles you are seeing, they are ribosomes. Okay? And then the rest are, you find a plasma membrane. Where is it? Where is it labeled? Where? It is here, the cytoplasmic membrane. And then you find a cell wall, and finally a capsule. So do you see that it has three protective layers? A capsule, which is usually silime, okay? Then under capsule we have there a cell wall, okay? Then under cell wall, now you have there a plasma membrane, which regulates what enters and what leaves. Then you have the pili, or if it is one, you can call it the pillars. Now these pili, they do perform different functions, where one of them is conjugation, exchanging the genetic materials, okay, the plasmids that are found within this nucleus. So there are these pili, some of them are modified to carry out conjugation, okay. Then you have this flagellum. Now this one is the one which brings about locomotion, okay, that it beats rhythmically to provide that propulsive force. It is going to move this organism forwards. Remember, it lives in water, okay, so it must be able to beat, this flagellum must be able to beat rhythmically to move this organism forwards. So you might think that this flagellum is an extension of the plasma membrane, but you need to study how it is attached. There's some interesting biology here, okay, so you need to. So if you want to draw, you can draw this one is for, wherever I usually put one for illustration, And I usually like these 3D pics. And then for drawing, so for drawing, you can draw this. That is literally how a bacterium is drawn. Then you have there the mesosome. Now, unlike the eukaryotic cells that do have mitochondria, okay, that do carry out respiration. Now, these ones, these bacterial cells, for them, they have membranes within themselves, within the cytoplasm, or an extension of the plasma membrane sometimes, which do carry out respiration. And these membranes are referred to as, it is a mesosome. Is that okay? Yes. So this is going to be your nucleoid region, and that's the circular DNA. Then this membrane, it is a mesosome, and then and so on and so on. We have talked about almost everything. So now, how about eukaryotic cells? Now, these eukaryotic cells, like I said, Eu means true, okay, and then karyotic means nucleus. So these ones, they are characterized by having a DNA. Now, whereas the other ones have a circular DNA, now these eukaryotic cells, for them, they have a linear DNA. And we shall study the structure of DNA, okay, in chemicals of life, and see how it is linear. So eukaryotic cells have a linear DNA, and the linear DNA is enclosed within a double membraned nucleus, or a double membrane organelle called a nucleus. It is enclosed there. It cannot move out. Neither can it cannot move out. It will always be embedded within a nucleus. But its embedment within a nucleus, or is it embodiment? Find out. Within a nucleus, it instructs, it controls all the physiology, okay, that takes place within the cell and the entire body. It is this DNA which controls that. However, it is static. It does not move. It cannot move out of a nucleus. However, it controls all the activities within a cell. So now in your mind, you should be thinking about how. Okay? How is this DNA going to do it? Okay? To give you a hint, the DNA is static, it doesn't move, but it has segments called genes. That these genes, they determine particular proteins to be formed. And they are the proteins that are formed from these genes, okay? They are the ones that do carry out. They are the ones that do catalyze most of the physiology in our body. So this DNA is going to be transcribed, okay, into an mRNA molecule, which is packaged by transport proteins within a nucleus, okay, and transported through the nuclear pore to the cytoplasm, to the ribosome, where now this mRNA is converted into a sequence of amino acids. And then after forming that sequence of amino acids, then you mold it, you modify it to give a protein of interest. And now it is that protein that will either be packaged and released out by exocytosis, or works within that very cell. So depending on the need. Okay? Remember how we discussed the signal transduction pathway. So you can relate. You can now relate. Then another characteristic is that they do have the membrane-bound organelles. The double membrane-bound organelles. For example, you find a mitochondrion. Okay? A mitochondrion within an eukaryotic cell, but you can't find it in a prokaryotic cell. You find a chloroplast, okay, within an eukaryotic cell, but not in a prokaryotic cell. You find nucleus, okay. You find in a complex array, okay, of these organelles of cell virtual, Golgi apparatus, endoplasmic reticulum, etc. And actually we shall study all those organelles. Okay? Yes. Cytoplasm is in cytoplasm in the region between plasma membrane and a nucleus. So this one is almost identical as prokaryotic cells. But the difference is here the cytoplasm is between the nucleus. Now it has a nucleus, that dense particle, that dense part within a cell. Okay? So it is between, okay, a nucleus and plasma membrane. Then are generally much larger than the prokaryotic cells. So if you compare sizes, these ones have large sizes compared to the prokaryotic cells. Okay? Then 80S ribosomes, I talked about this. So basically that is the structure, the 3D structure of an eukaryotic cell. Okay? And I usually like these 3D structures. Where you can see a nucleolus, that is that dense particle within a nucleus. Then you have a nucleoplasm. Okay? The whole of this space is a nucleoplasm. Then the whole of this is now a nucleus. Then this membrane is called a nuclear membrane. Okay? Now within this membrane, you have there tiny spaces. Okay? Tiny spaces. Actually they are regulated. They have protein molecules which regulate opening and closure of these spaces. Okay? So these spaces are called, yes, you can guess, they are called, good, nuclear pores. So now there are these pores which regulate what enters and what leaves this nucleus. Okay? Then away from the nucleus, then you have the nuclear envelope or the nuclear membrane. We talked about it. Then the nuclear envelope is continuous, okay, to the endoplasmic reticulum. It forms a continuous membrane system with the endoplasmic reticulum. Okay? And the endoplasmic reticulum is divided into two. We have the rough. Now, the rough endoplasmic reticulum, it is called rough because it has very many of ribosomes. Okay? So that's why it is called rough. Then you have the smooth endoplasmic reticulum. Now this smooth lack ribosomes. So that's why it is called smooth. Then the endoplasmic reticulum, it forms another continuous. Okay? Okay, there might be some space or sometimes it might be continuous. Okay? With another system of membranes called the Golgi apparatus. Do we have it? The Golgi apparatus. So that's how it is illustrated. But it is a system of stacked particles, stacked membrane, a system of membranes that does allow internal transport system. Okay? And packaging modifications, we shall see, we shall study all these ones in details. Then the cytoplasm, then you have the mitochondrion, okay? What else? The chloroplast, depending on which type of cell. If it is an animal cell, then don't expect to find this. But if it is a plant cell, then you find their chloroplasts. Okay? Then you may have centrioles, you may have very many virtuals, very many organelles within this nucleus. Then you have a plasma membrane, Okay, we can call it a cytoplasmic membrane, we can say plasmalemma, we can say cell membranes. It is still the same thing. Then if it is a plant cell, then you will add there a cell wall. Okay, so literally that is a basic view of a plant cell, and I promise we shall study all these organelles in details. Okay, and see what makes these organelles, how are they adapted to perform their functions, what are their functions, how can you describe the structure, and so on and so on and so on. Okay, so that is cell biology. You are now in a cell world. Okay, so this is a cell world. So basically we can talk about a plant cell and an animal cell, the panorama view. So that's why I told you I like these views, because they give you a clear picture of what a cell is. So on your left there is an animal cell, and on your right there is an animal cell. Now if you are to observe, they all have a nucleus. Okay, they all have this membrane system, the endoplasmic reticulum and the Golgi apparatus. They all do have. Okay, they all have mitochondria. Are you seeing? They all do have. They all have peroxisomes. They are needed, but they all do have. They all have a cytoplasm. Okay, they all have a plasma membrane. You can compare. Sorry, but there are some structures that are present within an animal cell that are absent in a plant cell, and I hope you should be able to spot some. For example, if let's demystify an animal cell, you cannot expect to find centrioles in plant cells. Okay, it is not by mistake that they are not there. You cannot expect to find flagella or a flagellum within a plant cell. However, there are some animal cells with flagella. For example, a sperm cell does have a flagellum. Okay, what is it for? Yes, that is it. You cannot expect to find microvilli, these extensions of the plasma membrane within a plant cell. You cannot, okay? And you cannot expect to find lysosomes. In other words, animal cells, but not plant cells, no lysosomes, okay? No centrosomes, these centrioles, and no flagellum, okay? But present in some animal cells, such as sperm cells. Now here they are saying that present in some plant sperms. Which plants? The mosses. Okay? But other higher plants, they don't have. Now with plant cells, you can see this is the basic structure. You cannot expect to find a chloroplast in an animal cell. Okay? That is some basic. You cannot expect to find a cell wall within an animal cell. Okay? You cannot expect to find a cell vacuum within an animal cell. Okay? And so you cannot expect to find these plasmodesmata, these ones, these, these junctions. However, if the epithelial cells, they can also form some gap junctions. Now there are those gap junctions which are now, remember epithelial cells, they are not innervated, they don't have blood capillaries. Okay? So exchange of materials is from a cell to another cell to another cell. So they must form those gap junctions which allow lateral movement of materials. So literally, that is a basic structure of a plant and an animal cell. Okay, so you should be in position to know. So you have an assignment. Okay, you have an assignment. So from what we have discussed, okay, oh, from what we have discussed, okay, are you able to compare an eukaryotic cell and a prokaryotic cell? And with comparison, I mean, can you give enough differences, okay, and enough similarities? Are you able? So this is another assignment that you're going to do. So I want you to compare for me an eukaryotic and prokaryotic cell. Give at least 10 differences and at least 10 similarities. Okay? Yes. So let me check for some work before we talk developed. Like the same way you were in P7, the science you studied. When you started O level, you studied some science. And now in A level, you are also studying some science. At campus, you study science. So that's actually how science was developed. Eh? From you start from scratch, and then you keep developing ideas up to where we are right now. Okay? So also a cell, little did we know about a cell, okay? As far as mid 18th century, little did we know, okay, about a cell. Why? Remember the other brief history I gave you about microscopes. Because these cells cannot be seen by our eyes if they are not aided. Why? Because our eyes have low resolution, low resolving power than what is enough to visualize these cells. Okay? So we cannot see the cells. But however, with continual improvements made to microscopes over time, okay, then it has helped us to discover new facts about cells. Okay? So with continued evolution of microscopes, then we discover new facts, and we are still discovering. Actually, this is some virgin field, some virgin land, where you have a lot of work to do. So you can do your research in cell biology and become a professor. You win Nobel Prizes and become a professor of cell biology. So the first man, we should even give a standing ovation to this man. This Robert Hooke is the one who began the science of cells. Okay? He's the one who was able to first postulate that there is a world of cells. Okay? That there is something called cells. Otherwise, before this man, I think, like, I don't even know. People used to think that organisms are just a mass of, I don't know. Okay? Because it is even these times when atoms were discovered. Okay? By using, was it carbon? That something called atoms. So people used to think that it's just, yeah, so you can imagine. So this Robert Hooke, he observed a piece of coke under the microscope, under his microscope. And you can Google about his microscope. You'll get amazed if you look at its picture. In fact, you get confused that how did he even use this? So he observed a piece of coke under a microscope, and he was able to see pores. Can you imagine? I hope these are the pores. So this is what Robert Hooke was able to see, okay, in a piece of coke. Piece of coke. Now he was able to see some pores. These ones. And then the man was amazed, and then he was like, what are these powers? What are they? Okay? But from now the modern world, from the modern understanding of cell biology, we now know that these powers were cells. So this is the picture that he was able to describe. He observed the powers and he was able to draw this. Okay? So do you see that this was some basic understanding, high understanding. Now from this research of Robert Hooke, then two German scientists, that is Matthias Schilden and Theodor Schwann. Now you don't need to cram these names, eh? No one will ask you. But I just incorporate them to make science, to give science that picture, to make it interesting. So Matthias Schilden and Theodor Schwann. I know you've ever heard of this somewhere. The Schwann cells. You know where the Schwann cells are?
Yes.
Good. Where do you find the Schwann cells?
The neurons,
In the neurons. That's good. So this is the guy.
motor.
This is the guy who discovered those Schwann cells, and he named the cells his name. So this Matthias Schilden and Theodor Schwann both studied cells, okay, of animals and plants. In fact, Schilden was studying plants, whereas Schwann was studying animal cells. But surprisingly, they discovered that these cells have some things in common and some things were different. So there were some significant difference between the two types of cells, the plant cell and the animal cell. Okay? However, they also had some things that were in common. Okay? So, from their research, these two were able to conclude that cells were not only fundamental to plants but to animals too. Okay? So, basing on the differences and the similarities, some structures are present in animal cells, others are present in plant cells. So they were able to postulate that probably these cells, they are fundamental units. They are fundamental structure units as far as plants and animals are concerned. Let me check if I still have some connection. Oh, you are still getting me?
Yes.
Oh, I had some notification which usually has something to do with network, eh? Yeah. So from that, they were able to postulate their own ideas of cells. Okay? But from those ideas, then we have been able to determine, to postulate. Okay, we've added these ideas together to bring about the modern view of a cell. And it makes a cell theory. Now, one, all living organisms are made up of cells. It's either one cell or more than one cells, many cells. Okay? And this one was postulated by Schwann and his friend. Then they were able to also postulate that the cell is a structure and functional unit of all living organisms. I think that's what we know right now. All the physiology. That's why I told you. All the physiology, all the life processes that enable you survive, they take place within a cell. So for you to understand science, then you must be able to know what a cell is, both internal and external, or organelles, and the processes that occur within a cell. Okay? So let's credit these guys. They did some good work to discover that really cells exist, and cells are the fundamental and structural units of living organisms. It was a breakthrough of science. Okay? And all these biology concepts, all of them, they do rotate around cell biology. And after the discovery of cells, then everything now became so easy to deal with. All cells arise from pre-existing cells by cell division. Okay? And this was Rudolph in 1858. Okay? So he carried out his study, and he was able to postulate this, that no cell, okay, no animal can produce cells. You cannot produce a cell in a factory, but rather these cells come from pre-existing cells. Now, to confuse you more, think about evolution. Where did that first cell come from? Is it true God created it? How did the first cell appear? Now that is evolution. Okay? To try to explain that, then you need the concept of evolution. And to be specific, that theory of chemical evolution. For me, I try to agree with that theory. Then, cells contain hereditary material, okay, which is passed from one cell to another cell by cell division, okay? And we shall study cell division and see how it occurs. Then all cells are basically the same in chemical composition. All cells are basically the same. Those minor differences are just differences to suit their surrounding. But basically, cells are the same. Okay? And then all energy flow, that is metabolism and biochemistry of life, occurs within a cell. So basically, these are the main views, okay, as far as the cell theory is concerned. However, that cell theory is not gospel truth. There are some exceptions. There are some deviations. There are some abnormalities from the cell theory. For example, number one, viruses. You know, viruses are neither cells, nor you can't rule out to be not cells. They are living organisms that are acellular. You can imagine. Why? They are obligate intercellular parasites that are not capable of reproducing on their own, on replicating on their own. So what they do is, they must do carry out replication under or inside a host cell using a host cell's machinery. So therefore, because of that, then they are considered as biotic, as to have life, but not organisms. Okay? They are not organisms, and they are not normal cells, because we have seen that a cell carries out its own physiology. Okay? But now these ones, you see. So they are exceptions from the cell theory. Number two, we have cynocytic, or you can say aseptate algae, such as this one, and then most of the fungi, most of them, okay, e.g. those ones in the zygomycetes, their bodies are continuous. It is just a continuous hyphae or a continuous strand, okay, with very many nuclei. Can you imagine? The nuclei are just embedded within this continuous strand, okay, and they are going to form different hyphae, okay, in such a way. So this condition, we call it aseptate, okay, or cynocytic. So now this one, where is a cell? Where is a cell? Hmm? Now are you going to say that this is a cell? Because it's where is it? Where does it begin from and where does it stop from? So now because of this, now we are in confusion. We don't know. They don't fit in the cell theory. Okay? So it becomes hard. Okay? To classify them under. You don't know which one is a cell and which one is not a cell. Our skeletal muscles, okay, they have cells which are elongated, you can imagine, with hundreds of nuclei within one unit, okay, of a skeletal muscle. You have very many nuclei, okay, within that unit of a muscle cell. Now, where are these nuclei coming from? Which one is a cell and which one is not a cell? Okay, so it becomes hard. So this term they are referred to as syncytia. Is that okay? Then we have giant algae. One algae which is just one cell but too big. Too big. I think I have some pic here. I don't. So it is too big. Okay? So it is an organism made up of one elongated cell. Imagine up to 100 millimeters long, but with just one nucleus. Okay? So this one does not fit within the cell theory. And finally, some tissues or organs contain large amount of extracellular matrix, e.g., the vitreous humor of the eye and mineral deposits of the bone and the xylem. Now, these ones, they have extracellular matrix but large deposits. And so it is expected to be within a cell, but now this is an organ, okay, with that matrix. Now it becomes confusing which one is which, okay? So this one also does not fit within a cell theory. So a question as far as cell theory is concerned?
No, not yet.
No question, eh? Good. So let's talk about a cell organelle. And I got amazed when I saw this pic. You can see you have the security gate here, okay? Then you have the cleaning crew. This one is responsible for cleaning. Then you have your kitchen, the powerhouse. Then this one, you have the skeleton. Then the control center, which is the nucleus. Then you have the industry, okay? The assembly unit. And then the work benches, the ribosome. So you have so the whole of this actually gives you a clear view, a clear picture of what happens within a cell. A cell is a complex array. It is complex than what you think. Okay? And what makes it complex? They are the physiological processes that this cell carries out. And because of those different processes, different reactions that occur within a cell, then the cell there must be compartmentalization. Okay? That each physiological process occurs within its own compartment called the organelle. So that's why you see powerhouse, security gate. Otherwise, if you now imagine if you don't have these ones, the kind of confusion that would be there. Okay? So this one introduces us to cell organelles. So they are the membrane-bound structures inside the cell's cytoplasm, which perform a special function. They do perform a special function. Each organelle performs its own function. And they are these organelles that keep cells moving. So the cytoplasm is usually light and it is it is composed, this should be O. It is composed of the following: number one, a soluble or soluble organic compounds such as sugars in the cytoplasm. Like I told you, the cytoplasm is not just an open space, okay? But rather, I can say a fluid-filled matrix which contains sugars, okay, amino acids in the cytoplasm. They must be there. Now, think about why sugars must be there. Think about why you should have there amino acids. Think about why you should have there proteins. And by the which proteins? Okay? Yes. I've given you an example. Enzymes. Which enzymes? And why should they be there? What do they do? Okay? And not just enzymes. You have there a complex. You have there different things. Not just enzymes, different proteins must be within the cytoplasm. Okay? To keep this cell moving. Then it houses organelles, I mean the cytoplasm. It houses the organelles. Okay? And these organelles include the nucleus. Think about it. What does it do? What is its structure? And why should it be there? Okay? The mitochondria. What do they do? What are their structures? Okay? The Golgi apparatus or Golgi body, endoplasmic reticulum, etc. And we shall study all these organelles one by one. Okay? You must be able to describe each organelle. You must be able to give the functions of each organelles and each organelle, and you must be able to tell us how a given organelle is adapted to its function. Okay? Then we have a network of fibers within this matrix, within this cytoplasm. Okay? So you can see this is a centriole. It forms a network of fibers. And these fibers, they include the microtubules and macrotubules and microfilaments. Now there are these fibers which make up the cytoskeleton. Think about what makes this cell to become like this, to become intact. Okay? So it is this cytoskeleton, the microtubules and macrotubules and macrofibers, they are the ones that constitute the cytoskeleton. Okay? And they are embedded within the matrix, the cytoplasm. So in a cell, the organelles of most importance, yes, they are all important, of course, but obviously, one must be more important than the other. So you have the nucleus, you have the mitochondria, you have the ER, the Golgi apparatus, the vesicles, and the vacuoles in case of plant cell. And others, you may talk about the chloroplast if it's a plant cell. You may talk about, not all organelles are important, however, some are more important. You can't say the nucleus is as important as a vacuole. However, they are all important, and we shall study each of these organelles. Any question for me?
No?
No question yet. Good. Now, this is where I'm interested in the cell membrane. And I want you to see there's some structure I want you to see. Okay, we shall see. So, you can also call it a plasma membrane. It is still the same thing. Or in some books, you may find plasmarema. Plasmarema. So, it is still the same thing. So, in case you find any of these ones, then you know that they are talking about the cell membrane. So it is found in all cells, all of them. No cell, as long as it's a cell, no cell does not have a membrane. And this membrane separates the interior of the cell from the outside environment, and it will regulate whatever is going to enter within a cell. Okay? So, this is a cell. It must have its own environment. And these cells, they are surrounded by the outside environment. And this constitutes the tissue fluid, for instance. Okay? So, whatever is here should not just cross into this cell. And also, whatever is in this cell should not just cross, okay, to outside of a cell. So, what determines that? You must have a gatekeeper. And a gatekeeper is our cell membrane. So, this cell membrane, it is the one which regulates, and it is the one which separates the internal, the inside of a cell and the outside. Now, it consists of a lipid bilayer. We are going to describe, we are going to see. Okay? So it consists of a lipid bilayer that is semi-permeable. Semi-permeable means it allows some materials to pass through, other materials fail to pass. So in other words, it regulates whatever is going to pass through. So it is a selective barrier that allows sufficient passage of oxygen, talked about this, nutrients, and wastes to serve the volume of every cell. So, for a cell to work, oxygen must be enough. So therefore, oxygen must easily pass through. The nutrients, you need nutrients for a cell. You need wastes because when this cell carries out the physiology, it will produce the wastes. So now these wastes must be able to move out of this cell. Otherwise, if they cannot move, then I don't know if the cell can continue surviving. So general structure, we shall talk about the structure in details. The one is of a biological membrane. Yeah, they can also call it this. So if they say biological membrane, it is still a cell membrane. It's a double layer of phospholipids. Yeah, like I told you, I like such 3D shapes. So basically that is a cell membrane. If I draw a cell like this, okay, and I say this is the nucleus, and this is the cytoplasm, and this is the cell membrane. If I magnify this, okay, this cell membrane, then this is what I get. So welcome to the cell biology. So if I magnify just this, you see how in O level they write to us? Just this is what I get. And it is a complex array, okay, of different molecules that interact together to form a cell membrane which surrounds a cell. Okay? Now, for you to see this, you may think it is as easy. They just woke up one day and they were like, yeah, the membrane is like this. It's not like that. People did people did research. Okay? People carried out work to come up to the present model of a cell membrane, which is called the fluid mosaic model. And so, let's talk about it. Now, as early as this, can you imagine? In 1935, nobody knew about a cell membrane. Maybe some few people who are just hypothesizing, who are just postulating some conspiracy theories about a cell membrane. Not until this, when Daniel Davison, Hugh Davison, and James Daniel carried out their research. on a cell membrane. Okay? And these two guys, they were able to come up with a Daniel-Dawson model of a cell membrane. Okay? And so when these guys observed a cell membrane, they were able to tell us that it was a structure like a sandwich. I hope you know a sandwich. You're going to see it. In which the hydrophilic proteins, hydrophilic. Now, I think this is a new term. Hydrophilic means water loving. Water loving, to love water. Okay? Then the opposite is hydrophobic. Hydrophobic means water hating. So these guys suggested that these hydrophilic proteins, they coat both sides of a pure lipid bilayer. So these guys were able, I will show you. So these guys were able to carry out some research, and they were able to say that it is a three-layered structure, of which you have two layers of proteins that do sandwich the pure lipid layer in the middle. And actually, we believed these guys. So they proposed that the plasma membrane is made up of three layers, a bimolecular layer of lipids in the middle, and a bimolecular layer is sandwiched between two layers of proteins. Okay? I'll show you the one what sandwich means. Now, the lipid molecules being set at right angles of the surface. Now, let me show you what that means. I hope you know this. This is a sandwich. Have you ever eaten this?
Yes.
Have you ever eaten this?
Yeah.
Oh, okay, then you must be lucky one. So these guys postulated that you can imagine that the plasma membrane is just like a sandwich. You see, this sandwich is a stack of different layers of carbohydrates. Eh? It is a stack. They are stacked together in different layers. Okay? So that is a sandwich. So they were also able to postulate that the plasma membrane is just three-layered structure, okay, with proteins on both sides. And here you have a layer of proteins and another layer of proteins, both on both sides, up and down. And then these proteins, they do sandwich a layer of lipids. Eh? Are you seeing this? So this is how these guys were able to postulate that is their plasma membrane. And by the way, if you remember the scanning electron microscope, this one just scans and gives you what is on the surface. And the picture or the photograph that it gives you is called a scanning electron micrograph. So these are the scanning electron micrographs. They were able to support their structure. And actually, this structure was believed for decades. The people believed in this structure. And if you want to write, if you want to draw with a pen, you can draw this. It is still the same thing. This is a layer of proteins. This is another layer of proteins. And this is a layer of lipids. So the proteins, they do sandwich, okay, the layer of lipids to form such a structure. So actually, people believed in these guys. They were the great guys by that time, because they had postulated something that wasn't there. Now, however, with further increase in research, okay, and one of them being the freeze structure. Is it freeze structure? Freeze structure. Here you freeze a membrane and then you break it so that you get the other first model that I showed on the first slide. They were able to see that the protein molecules. Now, the Daniel and Davson, for them, they postulated that the proteins just sandwich the top. They don't span through the membrane. But with further improvement in biology, in research, in techniques, then these two guys, it is the Nicole and what? Something. In 1972, they proposed that at least some protein molecules span through the membrane instead of being found on the outside by lipid layer. So that instead of sandwiching the lipid bilayer, some proteins do span through. Okay? So they were able to modify the Daniel and Davson's model to form what we call a fluid mosaic model. And we are going to see why it is called a fluid mosaic model. So they said that these guys, one of their conclusions was like, it is a bimolecular structure of lipids, okay? And so it is a bimolecular structure of lipid part of a plasma membrane. Now that this plasma membrane is made up of a bimolecular structure of phospholipids. And we are going to see those phospholipids. You have nothing to worry about. Then the proteins take the form of goblet dotted about here and there in a mosaic pattern. Okay? Unlike the Daniel and Davson's model, where proteins just do sandwich, okay, like the whole of the upper surface and the whole of the lower surface, they are proteins, and then these are lipids. Unlike the Daniel Davson's model of sandwich, now these ones for them, they say that it is just a bimolecular layer of lipids, that these are lipids. I don't want to talk about phospholipids before I introduce them. That these are lipids and they form a bimolecular layer. It's a layer of two. However, some proteins, if you observe from the surface, they form some dots. They are dotted, and the dots are in some mosaic pattern. There is no that uniform arrangement, okay? But they just form in a mosaic pattern, okay? If you observe from the surface. And that was, by the way, some good improvement, okay? And they also postulated that some of the globular proteins, in addition to forming this mosaic pattern, for them they can span through. Let me change some pen here. Some of the globular proteins they are attached on the surface, but also others penetrate into varying extents, and others they extend. But some of them extend right through it and stick on the other side. So you may form some of these proteins. Now, this mosaic pattern is observed when you observe on the surface. But if you try to observe inside, you see that some proteins can penetrate through the molecular bilayer. Okay? And at varying extents of penetration. But also we have some that just span through, okay, the bimolecular layer. They span through, okay, unlike these ones For them, they didn't talk about spanning through, and they didn't talk about these proteins penetrating through. So these ones, after freezing the membrane, they were able to separate, okay, the upper layer and the lower layer, and so they were able to see these integral proteins, these proteins that do span through, and it was an advancement, okay. And actually, this is the model that we still believe in, unless you do some research and come up with your model and convince us, then we shall believe you. So the membrane is then now thought to be far less rigid than it was originally supposed. Now, based on these guys, the Daniel and Davson, for them their membrane was rigid. And actually, even before these guys, they thought like a cell is surrounded by some rigid structure, hmm, that isolates the internal from the outside. But based on the research carried out by these guys, they were able to find out that the proteins, sorry, this membrane is far, far, far flexible. It is less rigid than how it was initially believed. Now we should be thinking about the rigidity. Where does the rigidity, sorry, the flexibility come? Where does it come from? What contributes to the fluidity of the membrane? Now there are two things. Okay? Number one, these phospholipids. Okay? And number two, these now in unlike bacterial cells and maybe plant cells, animal cells have cholesterol, and cholesterol is a steroid. And it is this cholesterol which maintains the fluidity of this membrane, both at high and low temperatures. And we are going to see. Now, experiments on its viscosity. Viscosity means how viscous, how fluid a membrane is, or any liquid is, suggested that it is it is of fluid consistency rather like oil. Like this, the way you see oil, eh? Like it's a viscous, the way oil is viscous, and so also this membrane is not as rigid as that. So molecules within this membrane are able to move, hmm, within this membrane. So considerable sideways movement of the lipids and proteins within this membrane. So we have some movement. It is not as rigid. It's not that if this protein is here, then it will always be here. It cannot move from this position to this new position. It's not that this phospholipid, because it's here, it will always stay here. No. There are some considerable movements within this membrane. So it is not as rigid as you think. Now that considerable movement is the one that contributes to what we call fluidity. Okay? So now because of that fluidity, because of that fluidity of the membrane, and because of the arrangement of these proteins, if you observe from the surface, not sandwiched, but they are just dotted in a mosaic pattern within the surface of the membrane. Okay? Then if we combine these two, then we shall form our model, the fluid mosaic model of a plasma membrane. So basically, that's how we describe the fluid mosaic model. The rest are you just talk about this. Maybe we have the glycoproteins, glycolipids, the cholesterol, and so on. So that is the fluid mosaic model. And these guys did some good work. And up to now, we still believe them. So basically, that is what they postulated. Okay? that if you observe, it is a double layered structure. You can see this is one layer, this is another layer of phospholipids. Okay, it is a double layered structure of phospholipids. Now within the phospholipids, you have their protein molecules that do span through, or that do penetrate through the phospholipid bilayer at different length, where you have those ones which are intrinsic and we have those ones which are just on top at the surface. We call them extrinsic proteins. Okay, then we have those ones which are integral proteins. They just span through, okay, the whole bilayer. And by the way, they are the ones that form a channel. And by the way, when we are talking about coordination, this one is going to be key. These proteins, they are key. That's why I wanted us to first study cell biology before we talk about transmission of an impulse. Then that is a phospholipid. Let me charge. So that is a phospholipid, this one. And if you are to study the structure of a phospholipid, it has a head with two fatty acid chains. Okay? So a normal triglyceride, it has a glycerol with three fatty acids. But now, information of a phospholipid, the fatty acid, one of the first fatty acid is replaced by phosphoric acid. Okay? So that you end up forming a glycerol which is attached to this fatty acid and a phosphoric acid here to make the head. And so these two, they are going to form a tail or tails. So this is the phospholipid tail, this is another phospholipid tail, and all of this forms a phospholipid head. So what you are seeing, this one, it is glycerol together with phosphoric acid. Then this one, this is just a long chain, a long hydrocarbon of a fatty acid, of fatty acids. Now since these hydrocarbons, if I just write this one, it is carbon, hydrogen, carbon, hydrogen, carbon, hydrogen, carbon, hydrogen. This one therefore becomes non-polar. Okay? And so being non-polar, it is hydrophobic. It is water hating. Okay? But the heads, because you've added more OH groups, you've added phosphoric acid, and phosphoric acid is H3P4, PO4, this one. So you've added more polar groups, but also the glycerol also has some oxygen. Okay, you've added more polar groups. Then these polar groups, they are going to increase the polarity of the head. And so this head ends up being, yes, hydrophilic. That when these phospholipids are oriented within water, let me erase over. So you can read about the hydrophobic interactions and hydrophilic interactions. So if you have various phospholipids within, and they are oriented within a solution, and that solution contains water, the phospholipids, the phosphate heads, these heads, they are going to orient to the side that has more water because they are water loving. And these tails, they are going to orient to the side that is away from water. Okay? So, as a result, because of this property, then these phospholipids are able to form a biological membrane, a plasma membrane. Because think about this. This is extracellular, outside face, and this is inside. And inside you have where the cytoplasm, which is filled with water. But also outside you have there the intercellular space, which is also filled with water. Okay? So the heads that are water loving, they will orient themselves to the side where there is water. Even you can see this. They are orienting outwards to the side where there is water. But the tails, which are water hating, they are orienting themselves inwards. So because of this property, then you'll be able to form a plasma membrane. Then you have there short chains of carbohydrates. You can call them glycans. Okay? And the carbohydrate that is attached on a protein is known as a glycoprotein. Okay? Glycoprotein. Then a carbohydrate that is attached on a lipid, it is known as, for example, now like this one. I hope this one is attached on a lipid. But if I want to illustrate, I can attach it here. So this is a phospholipid. So I can attach a carbohydrate by glycosylation. That's the process. So this carbohydrate, we call it a phosphor, so the glycolipid. Why lipid? Because it is a carbohydrate that is attached on a lipid. Whereas this one, it is a carbohydrate that is attached on a protein. So that is basically a basic understanding of a plasma membrane. Is that okay? So for issues of drawing, you can draw this. But for the issues of understanding, giving a clear view, a clear picture, then this one. So now these proteins, let's talk about proteins and then we call it a day. Because they are the ones that we are going to use in
Teacher,
coordination. Yes.
first take back the other slide. I want to screenshot the.
Let me raise these ones. Yes, you can now screenshot.
Thank you.
Yeah, you're welcome. Now these proteins. Now let me first take you back also. Now it is not easy to cross this phospholipid bilayer if you are polar. Remember in organic chemistry we said that polar molecules dissolve polar solvents dissolve polar molecules, and non-polar solvents dissolve non-polar solvents. So now,
Sorry.
if you are polar, for example, glucose, for example, water, the ions, sodium ions, potassium ions, and so on, it is not easy to cross this membrane. Not easy at all. Why? Yes, you might cross the head. You might cross this head because it is polar. But you cannot cross this hydrocarbon. It is a long chain. Moreover, two of them. Hmm? Two of them. So when, let's say, sodium ions reach here and they reach the head, if they reach this non-polar, this layer, which is non-polar, they cannot cross. So, therefore, if you cannot cross, then how are you going to enter into the protein? Then you must be aided. You must be helped. However, I don't prefer those two words. You must be aided to enter into the plasma membrane or come out. And that process, we call it facilitated diffusion or active transport. Now there are these protein molecules, okay, that provide that channel, that pore, where polar molecules are going to cross. Now yes, they are specific. However, some proteins, they do provide a pore, like how you see this. They are transmembrane, so it just opens, okay? Some are gated. Now we are going to see the gated channels in impulse transmission. They are so important. Some are gated that in case of a stimulus, then it opens and the ions enter. Okay? The ions enter. Then after, it closes again. However, some they are more of enzymes. They are enzymes. Okay? Within this, they are more of enzymes. That what happens, it binds to a substance, and then after it binds, then it changes configuration, okay, and it pours that substance to the other side. You can we call that one ping pong, the ping pong model, so you can look about it and see. So in other words, there are different ways, okay, of transporting materials, but it's not just transport. You see, there are also recognition sites, hmm, that a protein, let's say hormone is going to bind, okay, and then after the hormone has bound, it induces processes, it induces reactions within a cell. You remember the other signal transduction pattern. So therefore, these proteins must be there. Actually, some they don't just form transmembrane, but some actually do span. Hmm? They do span through. Like this is your protein. It has the external surface, and then it spans through the membrane, something like 7 times. Okay? And then this is another site where the intercell, now the inside proteins will attach. Okay? So now these proteins, they are the ones, now if a hormone attaches here, then this protein undergoes some configuration to bring about changes here, which will induce a cascade effect. So these proteins, they do have a lot of function. Actually, there is somebody can set a question that discuss the proteins or the function of proteins within a biological membrane, and you discuss. So for such a question, then that's how you attack. They provide support, first of all, to the membrane, okay, but as if that is not their primary function. How do they provide support? They provide attachment sites for the microtubules. Remember what we said, that the cytoskeleton is made up of a centriole, actually centrosome, which is, there are two centrioles, and these centrosomes, they form microtubules. Okay, so now these microtubules, they attach on these proteins. Okay, they attach on a protein molecule within a plasma membrane. Okay, to maintain the cyto to maintain the cytostructure, to maintain this structure. Okay, and also they provide, they form interactions, both the hydrophobic and hydrophilic interactions between the proteins and the phospholipids, which regulate, which maintain the structure of a plasma membrane. Then we have carrier proteins. Now these ones, they do transport molecules against their concentration gradient, and in the process, energy is used. Okay, and where do you get the energy from? Active transport. So it means that some proteins, they work as enzymes. that will break down, that will hydrolyze ATP, okay, to ADP, and the energy that is evolved is going to be used in active transport. So all those are proteins within a plasma membrane. The channel proteins, they just provide a pore where polar molecules will just pass. So some are gated, they have a gate which opens and closes, but others they are not gated, they are just ever open. Okay? So materials will always diffuse in and out based on their concentration gradient. They are receptors, okay? They are receptors for the chemical messenger, such as hormones. So they have specific configurations, specific shape, which complements these hormones, that when a hormone comes, then it binds in the shape, in the site, the active site of a protein, and then induces a cascade effect, okay, which will lead to a change in physiology. Then some serve as enzymes for energy coupling reactions, e.g., to produce ATP. I want you to look at this. I want you to look at this. So this is an ATPase. ATPase. Now, what does this ATPase do? In respiration, I wanted some space. In respiration, there is what we call chemiosmosis. Chemiosmosis or chemiosmotic model. It is also in photosynthesis of production of ATP. Okay? You produce ATP by chemiosmosis, and this is a protein that is involved. How? It uses an electrochemical gradient, okay, that is produced by the difference in hydrogen ions between the intermembrane space and the matrix, okay, of the mitochondria. We shall study this as far as respiration is concerned. Say that if you have a high concentration of hydrogen ions in the intermembrane space than what is in the matrix, then at any time t, these hydrogen ions will always tend to diffuse. But because they are polar molecules, then they can't pass through this phospholipid bilayer. Here, they cannot. So you have there a specific channel protein or a specific protein that will provide a channel where these ions will diffuse through. Okay? Now, because of this difference in concentration gradient, as these ions are passing through this channel, then this protein is going to use that concentration gradient, that electrochemical gradient, okay, to add an inorganic phosphate on ADP to form ATP. And so this is how energy is formed by chemiosmosis. So now this, if you don't, you have a question? So if you don't have these proteins within the plasma membrane, then there is no way how you're going to form the energy. So we are going to stop here for today, unless you have a question.
No, no, no.
No question, eh?